My name is Hannah Brink, and I'm an undergraduate student in Dr. Todd Sulchek's BioMEMS and Biomechanics lab.
This summer, I was honored to have received the President's Undergraduate Research Award for my project on investigating the biomechanical effects of chemotherapy on leukemia cells using atomic force microscopy, or AFM. AFM measurements consist of a microscopic cantilever that temporarily indents individual cells, recording the approach and retractive forces of the cell body. Together with my lab partner Bryn McFarland, we have been maintaining two leukemia cell lines, K562 and HL60. Our research this summer focused on our K562 cell line, which is chronic myeloid leukemia, a form of blood cancer that is mostly seen in adults and is caused by a gene mutation. We were expecting the leukemia cells to increase in stiffness when exposed to chemotherapy, and were curious whether and how differing amounts of chemo added to the leukemia cells would affect their biomechanical properties. To answer this question, we treated our K562 cells with varying amounts of daunorubicin, a common leukemia chemotherapy treatment. After we performed our control experiments, chemo experiments 1 and 2 consisted of treating the K562 cells with what would be a normal amount of chemotherapy relative to the concentration of cells. The third experiment simulated a half treatment of chemotherapy, and the fourth experiment simulated a double treatment of chemotherapy. As can be seen in these box plots, with the untreated control K562 cells as a baseline, as we increase the concentration of chemotherapy, the average elasticity measurements of the cells in the sample also increased. This suggests that the ratio of cells that were accepting the chemotherapy, proceeding through apoptosis and thus stiffening, increased as the amount of chemotherapy they are exposed to increased. This poses the question, why is it that some cells accept chemotherapy while others appear to be chemoresistant? Next semester, we will be expanding our experiments to our HL60 cells to assess for correlation between the stiffness, size, and shape of chemotherapy-treated cells. We will use this mechanics data as selective criteria for a microfluidics experiment to sort and study the cells that display chemoresistance. I am very grateful for the opportunity to pursue these possibilities, and Bryn and I look forward to continuing our research on this topic in Dr. Sulchek's lab.